Hello friends, today we will discuss on the topic extraction of volatile oil, and this slide covers a small introduction to the volatile oil, various methods used, its applications, advantages and disadvantages. So after this lecture, you will be able to select a proper method for the isolation of volatile oil from a particular part of the plant material. The various extraction methods of volatile oil includes distillation, expression, extraction with volatile and non-volatile solvents, advanced phytonics method, supercritical fluid extraction, and enzymatic hydrolysis. The most common method used for the isolation of volatile oil from the plant parts is the distillation method. So what is distillation? Distillation is the process of conversion of the water molecule into a vapor form, which in turn condenses to form the water back. So this process is known as the distillation. And under distillation, there are three different methods of distillation. First one, water distillation, water and steam distillation, then steam distillation. So let us see what is water distillation. So water distillation is otherwise known as hydrodistillation, and it is most suitable for plant materials which are having active constituents which are not degraded by boiling up to 100 degree Celsius. That means those constituents which are not degraded when we are boiling the plant parts to a temperature up to 100 degree Celsius. And Clevenger's apparatus is one of the apparatus used for hydrodistillation in the laboratory level. So in the distillation method, we are immersing the plant material, usually the dried plant parts. It can be the leaf, then flowers, barks, etc. This is completely immersed in water, and then it is boiled by applying heat directly or indirectly. And once we heat the plant material which are immersed in the water, what happens? The water will vaporize, then volatile oil also will vaporize, because the volatile oils are highly volatile in nature even in the room temperature. So on the application of heat, what happens? The water as well as the volatile oil is converted into the vapor stage, and this is passed on to the condenser. And what is the function of condenser? It condenses or cools the water vapor as well as the volatile oil vapors. So these vapors will condense and then fall into a graduated tube. That is, we will get water as well as the volatile oil or condensation of the vapors. And the volatile oil being lighter, it will float on the top of the graduated tube, whereas the water which is present in the lower part of the graduated tube will return back into the round bottom flask, and the process is repeated. Since it forms two different layers, we can easily separate the volatile oil from the water. Example of volatile oil which is separated by hydrodistillation method include the lemongrass oil, turpentine oil, etc. And coming to the advantages of this method, it is a very simple method. We can easily understand it. That is, just by boiling, it will be converted into a vapor, and again on cooling, it will return back into the liquid stage. Okay, so this is a very simple method, and it is, since it is a simple method, it is cost effective, and no solvents are used in this particular method. So toxicity problems are not there because we are using only water, no other organic solvents. And coming to the disadvantage, even if they have some disadvantages like cost effectiveness, simplicity, and use of no solvents other than water, The main disadvantage is incomplete extraction. That means when we are boiling the plant part in the water, we cannot isolate the complete amount of volatile oil which is present in the plant material. After extraction also, some of the volatile oil may be left behind in the plant material. Then next one, that is the chemical alteration. That means the volatile oil is a mixture of so many chemical constituents which includes mainly the terpenes, aldehydes, esters, etc. So what happens is when we are boiling the plant part, there are chances for the active constants like esters and aldehydes which are present in the volatile oil to get degraded or hydrolyzed. For example, the esters, when we are boiling at a higher temperature, they may partially hydrolyze, and very sensitive substances like aldehydes, they tend to polymerize. So these are the two important disadvantages of this hydrodistillation method. Water and steam distillation. This method is used for the isolation of volatile oil from fresh as well as dried plant parts. And it is an ideal method for the separation of active constants that undergo degradation when boiling to 100 degrees Celsius. That is mainly those constituents which are degraded by hydrodistillation methods. Examples of some volatile oils separated by these methods are clove oil and cinnamon oil. Now coming to the process, in water distillation, we have seen that the drug is completely immersed in water. That is why more degradation of active constituents taking place. But here we are not immersing the plant material in water, but we are keeping these plant materials on a perforated grid above the water. So in a closed system, the water is placed at the bottom, and above the water a perforated grid is placed, and in the grid we are placing the fresh or the dried plant material. Now we are heating the water. So on heating the water, what happens? The water is converted into the vapors, and the vapors is made to pass through the plant material. So in the plant material, the volatile oils are present, and the volatile oil, you know the property of volatile oil, they are highly volatile in nature. So while passing the steam or the vapors through the plant material, what happens? The volatile oil, which is very volatile in nature, will vaporize. So the steam will carry the volatile oil vapors into the condenser. So what is the function of condenser? It just condenses or cools the vapor part. So here we'll have the vapor of volatile oil as well as the water vapor. So while passing the water vapor and the volatile oil vapors through the condenser, what happens? They will condense or cool to form the water and volatile oil, and it is made to fall onto a separator. So in the separator, you can see there are two parts. One is the dotted lines which represents the water, and over that you can see the oil separated, where we'll get the volatile oil. Usually most of the volatile oils are lighter than water. That means the specific gravity of the volatile oil is less than that of the specific gravity of water. So since the specific gravity is less, the volatile oil will float on the top of the water. But there are certain exceptions like cinnamon oil. It is heavier than water. So what happens if the oil is heavier? The oil part will be on the bottom of the separator, whereas the water will be on the top of the separator. Anyway, we will get the oil as well as water into two separate layers and can be easily separated. So coming to the advantage, the decomposition is less. It requires less fuel, and the yield of volatile oil is more when compared to the hydrodistillation process. One of the major disadvantages of hydrodistillation is incomplete extraction. And here the advantage is we'll get more volatile oil when compared to the hydrodistillation process, and the degradation of active constituents due to the direct heat is also less in case of water and steam distillation. And coming to the disadvantage, in case of high boiling volatile oils, for complete vaporization, we require a large quantity of the steam, and hence the time may be more. So there are two major disadvantages. One, large quantity steam may require. Second one, it may require the more time when compared to the hydro distillation. Steam distillation. It is one of the methods used for the isolation of active constant, that is the volatile oil, from fresh plant material. And it is an ideal method for heat sensitive and high molecular weight oils. And coming to the procedure, in the diagram you can see there is an external boiler where water is placed inside, and when heat is applied, what happens? The water in the external boiler get vaporized, and the vapors from the external boiler is injected into this another still, in which there is a perforated plate over which the plant material is placed. So what happens when the steam is injected into the still from an external boiler? The steam, while passing through the plant material, carries the volatile oil. That means the volatile oil is present in the plant material, and while passing the steam, what happens? The volatile oil, which are highly vaporizable in nature, will get vaporizes, and this steam will carry the volatile oil vapor, and it will pass to the condenser. And what is the function of condenser? It condenses the vapor into the liquid form. So here in the condenser we have the water vapor as well as the volatile oil vapor. So on condensation, what happens? We'll get water as well as volatile oil. So this, after condensing, will made to fall onto a separator. We'll get the water as well as the volatile oil. And the volatile oil, being lighter, will float on the top of water, and they will form two separate layers and can be easily separated. As I told in the previous distillation methods, some of the volatile oils are heavier than water, so those oils will form a lower layer and the water will be on the upper layer. Even though they are on the upper layer, since they are formed as two different layers, we can easily separate them. And coming to the advantage, the yield is high when compared to other distillation methods, and it is a simple apparatus, and hence we requires only less labor. And one of the disadvantages is degradation of active constituents due to excessive heat. And volatile oils like rose oil is mainly isolated by this method. So these are the three different distillation methods. Once more I will say, the first one, that is the hydrodistillation. Here we are immersing the plant material in water. Second one, that is the water and steam distillation. So in a still, we are placing the water, and over which we are placing a grit where we are placing the plant material. The third one, that is a steam distillation. So in the steam distillation, a steam is generated in an outside still, and this generated steam is made to pass through the plant material, which is kept on a perforated vessel. Next method is expression. This is mainly used for thermoliable drugs. That means those drugs which are degraded on application of heat, or those drugs which degrade on contact with the heat. So for those substances, we can use this expression method, and also for those plant materials which contain less amount of volatile oil. And this expression method is mainly used for the separation of volatile oil from citrus fruits like lemon, orange, etc. And by this method, we can preserve the natural fragrance of the oil. And expression method are of different types. That is, first one, Useley method, sponge method, and rasping method. So once more we'll see, expression method is mainly used for those drugs which are thermoliable in nature. That means those drugs which undergo degradation on contact with heat or on subjected to heat. And mainly it is used for the separation of volatile oils, which is present in very lower quantities in plants. And mainly this method is used for the separation of volatile oils from citrus fruits, and this method preserves a natural fragrance. And under expression we have three different techniques, that is the sponge method, rasping method, and Useley method. Under expression we have the sponge method. So as I told you before, the expression method is mainly used for the separation of volatile oil from the citrus fruits, where the volatile oil is present in lower quantities. So here the citrus fruits are first taken, they are properly washed to remove the dirt, and then they are cut into two halves, and the juice is completely removed by squeezing. After the removal of the juice, the rind is taken and it is turned inside out by means of the hand. And it is also squeezed to rupture the oil glands which are present on the surface of the peel. You know, when we are squeezing the peel of the orange or the lemon, we can see the volatile oil coming out. So the method used here is the similar one. While turning the rind or the peel inside out, what happens? The volatile glands present in the surface of the peels will get ruptured and the volatile oil is released. So the oozed out oil is then collected on a sponge, and when it gets saturated, the sponge is squeezed onto the beaker or a vessel. So what happens, we'll get some juice as well as the volatile oil, because when we are squeezing, even though we are removing the juice from the citrus fruits, there is chance of small amount of juice to come into the volatile oil. So this oil is allowed to stand for some time. So what happens? The volatile oil, being lighter, will float on the top of the surface, leaving behind the juice on the bottom layer, and it can be easily separated. So this is the sponge method for the isolation of volatile oil. The next method under expression is Useley method. So here you can see a specially designed apparatus, which is nothing but a large bowl which is meant for pricking the outer surface of citrus fruits. So this method is mainly used for isolation of volatile oil from citrus fruits. The inner layer of this apparatus, you can see numerous pointed needles, just enough to penetrate the epidermis. And the oil glands in the citrus fruits are present in the epidermal region. So these needles, they will penetrate the epidermis and break the oil glands to release the volatile oil. And the lower portion serves as a handle and the receiver for collection of oil. And the method here is we are placing the freshly washed lemon in the bowl and they are rotated repeatedly in order to puncture the oil glands and to discharge the volatile oil. And the remaining oil which is stick on to the needles are collected by a spray of water. And the resulting oil water emulsion is separated by centrifugation process. Another method under expression is rasping method. It is also meant for isolation of volatile oil from citrus fruits like lemon. The outer region of the pericarp of citrus fruits contain the oil glands. So in this method, first we are grating the pericarp of the citrus fruits and these raspings are kept in a horse hair bag and they are squeezed or pressed strongly to get the volatile oil. Next method for the isolation of volatile oil is extraction with volatile solvents. In this, we are using various organic solvents or volatile solvents like solvent ether, petroleum ether, benzene, etc. And coming to the process, we have to select an analytical grade purified solvent and this solvent is treated with the plant material from which the volatile oil has to be extracted. And after extraction, we'll get a extract and from the extract we have to remove the solvent, that is by evaporation process, and the extract is actually a mixture of vegetable wax as well as the volatile oil and this is known as the essence concrete. And from this essence concrete, we have to remove the wax. This is mainly done by shaking with alcohol, then cooling to 0 degrees Celsius. What happens is the wax gets solidifies and it is separated by filtration process. Now we have a mixture which contains the volatile oil as well as the alcohol and the alcohol is removed from the volatile oil with the aid of vacuum and we'll get the pure volatile oil with natural odor. And advantage of this method is uniform temperature of usually 50 degrees Celsius is maintained throughout the process and as a result we'll get the volatile oil with more natural odor. Next method of extraction of volatile oil is extraction using non-volatile solvent. Under this, the first method is enfurage method and it is mainly used for the isolation of volatile oil from flower drugs like rose, jasmine, etc. Here, fat is thinly layered on both sides of the glass plate which is supported on a rectangular wooden frame which is known as chassis. You can see the chassis in the picture. And several chassis are placed one above the other sandwiching the flowers between the two layers of fat. So first the fatty layer is coated on the glass plates and furrows are also made on the fatty layer in order to increase the area of absorption and once the fat is coated on the glass plate then the freshly picked flowers are thinly layered on the fatty layer and several chassis are placed one above the other sandwiching the flowers between the two layers of the fat and the loaded plates then are allowed to remain for 24 to 28 hours. During this period, the fat absorbs the essential oil released by the flowers. And once the flowers are exhausted, they are removed and this process is known as defurage process. and the exhausted flowers are replaced with the fresh ones. Reversing of glass plate called pattage is also done in order to maximize the percentage of absorption. And once the fats get saturated, the fat is scraped off and then it is treated with or extracted with alcohol. So what happens is the volatile oil get dissolved in the alcohol and the fat which is left out is filtered and removed. Also we can chill the alcohol to separate the waxy or the fat material. And now we'll have the alcohol in which the volatile oil is dissolved. So we have to remove the alcohol. This is done by different methods. First one, fractional distillation under vacuum at 0 degrees Celsius or evaporation under vacuum at 0 degrees Celsius. And also by treating this alcoholic extract with water and then with saturated sodium chloride, what happens is the oil will get separated from the alcohol. So these are the three important methods used for recovering the volatile oil from the alcoholic extract. Here we are not using the heat, so the volatile oil, whatever is obtained after leaving the alcohol, is highly concentrated and with a pure fragrance, and this is known as the absolute. So one of the major advantage of enfurage method is here heat is not used, so natural odor can be retained and the degradation can be prevented. Next method is pneumatic method. This is also one method under extraction of volatile oil using non-volatile solvents, and it is a modified form of enfurage method. Here a current of warm air is passed through the flowers. So what happens, because of the warmness of the air, it will vaporize the volatile oil present in the flowers, and this air laden will carry the essential oils or the volatile oil, and it is allowed to spray onto the melted fat in a closed chamber. So while the air laden is made to fall onto the melted fat, what happens? The fat will absorb the volatile oil. And from the fat, the volatile oil is recovered by treating the fat with alcohol and then chilling it. So the volatile oil gets dissolved in the alcohol and the fat is remain as a solid material and can be easily removed. And the volatile oil from the alcohol can be separated by evaporation under vacuum at 0 degree Celsius, which is very much similar to the enfurage method. Maceration is another method under extraction of volatile oils using non-volatile solvents. This is also one of the methods used for separating or isolating the volatile oil from flower drugs. So the flower petals are taken and they are gently heated in the molten fat. They are stirred frequently until complete exhaustion takes place. And once the flowers are get exhausted, they are strained and squeezed. And the fat, when they reach a desired concentration, it is removed and it is treated with alcohol. So what happens? The volatile oil present in the fatty layer get dissolved in the alcohol, then it is chilled. So during chilling process, the fat remain as a solid material, whereas the volatile oil will remain dissolved in the alcohol. So the fat which is present as a solid material can be easily removed by filtration process. And now we have the extract which contains the volatile oil and the alcohol, and the alcohol can be separated from the volatile oil by distillation under vacuum at 0 degree Celsius. So you will get a pure volatile oil. We have already seen the various traditional methods of extracting volatile oil, which includes the distillation, expression, and extraction with volatile and non-volatile solvents. Now we'll move on to the various modern methods for extracting volatile oil. These include supercritical fluid carbon dioxide extraction, microwave assisted extraction, ultrasound assisted extraction, and molecular distillation. Coming to the advantage of these modern methods over the traditional methods, the first one is the quality. So by these modern methods, we'll get highly pure oil with less chemical degradation. Second, efficacy. So the yield of the volatile oil obtained by this method is very high when compared to the traditional methods. And the third advantage is sustainability. That is, there is a reduced solvent waste and low energy consumption in all these modern methods of extracting volatile oils. Next, about the modern methods of extraction under this, the first one is the microwave assisted extraction method. Here we employ the microwave radiation directly to heat the moisture within the plant cells, which causes the rupturing of the various oil glands to release the oil. Also, solvent-free microwave extraction is available, which is more efficient, fast, and eco-friendly. The second one is the ultrasound assisted extraction method, where we are using the ultrasonic waves to create a cavitation in the solvent, which in turn causes the rupturing of the plant cells and increase the oil yield in less time. Molecular distillation or short path distillation. This distillation operates under high vacuum conditions and also low pressure, which allows the distillation of heat sensitive compounds at much lower temperature, preventing the degradation. So we can isolate the volatile oil without any degradation of active constants. Enzyme assisted extraction. Here we are using certain enzymes like cellulose or pectase to break down the plant cell walls, which in turn causes the liberation of volatile oil easier. Then modernized pressing or USLA method. Here the method is same as USLA, but the oil is separated from the juice by means of centrifugation process. So we will get a more pure oil. Then last one is the supercritical fluid extraction. Here we are using the carbon dioxide fluid to extract the volatile oil from the plant material, and here we are using very low temperature, and hence we'll get a high quality oil without any heat degradation. Now let us see what is a supercritical fluid. So for this we should know what is a critical point of a pure substance. It is the highest pressure and temperature at which a substance can exist in vapor liquid equilibrium, and at a pressure or temperature above this particular critical point a single homogeneous fluid is formed, and this is known as the supercritical fluid. And this supercritical fluid possesses properties of both gas as well as liquid. And in supercritical fluid carbon dioxide extraction, we are using carbon dioxide as a supercritical fluid. And why we are preferring this solvent? Because it is non-toxic, non-inflammable, non-corrosive, then it is chemically inert. That means it does not react with any other chemical constants which is present in the plant, and it has got a low critical pressure and low critical temperature. So degradation is also avoided. Then it is easily available, cost effective, and it has got environmental acceptability. That is why we are using supercritical fluid carbon dioxide for extraction of volatile oil in supercritical fluid carbon dioxide extraction. Now let us see what is supercritical fluid carbon dioxide extraction for the volatile oils. So in the picture you can see a carbon dioxide tank where we have the fluid carbon dioxide. Then it is pumped into the plant material which is kept in the extractor. So here the pressure is maintained and the temperature is also very low, and the plant material is protected under the blanket of carbon dioxide. This is mainly to prevent the oxidation of the drug. So from the tank, the fluid carbon dioxide is pumped onto the extractor, and while passing the fluid through the plant material, it extracts the volatile oils. Then it is brought into a separator where the pressure is released. So what happens? Only under pressure the carbon dioxide exists in the form of a fluid. When pressure is released, the carbon dioxide will convert into the gaseous form, leaving behind the volatile oil in the collection vessel, and the gas is again brought back into the carbon dioxide tank where it is again pressurized to convert the gas into the fluid, and the process is repeated several times. Let us see what are the advantages and disadvantages of supercritical fluid carbon dioxide extraction. So this is one of the most modern method used for the extraction of volatile oils. Here we are using the fluid carbon dioxide as a solvent, and the fluid carbon dioxide penetrates very quickly and extracts compounds up to a molecular weight of 400. Second advantage is, since it is operated at a temperature of 10 degrees Celsius, it avoids the degradation of chemical constituents due to heat. And the third, the yield and fragrance of volatile oil is more when compared to the other traditional extraction methods. For example, the yield of piper nigrum oil is about 2.8% in supercritical fluid extraction, whereas it is only 0.6% in steam distillation. Similarly, the fragrance of rose oil is more when obtained by this method. Coming to the disadvantage, since it is carried at a high pressure, it makes this process complicated and potentially dangerous when compared to the other traditional methods of extractions of volatile oil. This is the picture of a commercial scale supercritical fluid carbon dioxide extraction plant. Advanced phytonic method. This is also one of the most modern method to extract the volatile oils. Here we are using the phytosol as a solvent, and a sample chamber is filled with the plant material to be extracted. An aerosol can containing the pressurized phytosol is applied onto the sample chamber, and the whole system is kept under pressure to keep the phytosol in the liquid form. So here what we are doing is, we have a sample chamber in which the plant material from which the volatile oil to be extracted is kept, and there is another aerosol can where we have the phytosol under the pressurized condition, and then we'll spray the aerosol containing phytosol into the sample chamber. So while spraying the phytosol will move through the plant material which extracts the volatile oil, and this phytosol is then led into a collection vessel. And in the connection vessel, the pressure is disconnected or released. So what happens is, the phytosol is vaporized, leaving the extract material, that is nothing but the volatile oil. And the vaporizing phytosol is again collected and recycled. It is almost similar to the fluid carbon dioxide extraction, where the carbon dioxide gas is kept as a fluid form in the pressurized condition. Similarly, here also the phytosol, which is a gaseous form, is kept under pressurized condition in the form of a fluid which is passed through the plant material to extract the volatile oil. And what are the advantages of this method? It is more environment friendly, and it has the ability to modify the solvent to improve the extraction capabilities, lower capital cost, and much safer operation because it is carried out low pressure and temperature when compared to the supercritical fluid extraction where we have a very high pressure which makes it a complicated process, and it is also non-inflammable in nature. So these are the advantages of advanced phytonics method. In some plants, the volatile oil is found in a combined form or it may occur as glycosides. In these conditions, first we have to release the volatile component from the glycosides by means of enzymatic or water hydrolysis and followed by steam distillation to separate the volatile component. For example, in wintergreen, the volatile component methyl salicylate is present as guanidine. So we will do the hydrolysis and then the methyl salicylate is separated and this is isolated by steam distillation process. Similarly, in GM urbanum, eugenol is in a combined form that is known as the gene, and in bitter almond, benzaldehyde is found as amygdalin. Another example is in black mustard, the allyl isothiocyanate, which is an volatile component, is found in the form of sinigrin. So in all these compounds, after enzymatic hydrolysis or water hydrolysis, we can separate the volatile component, which is followed by steam distillation to isolate the volatile component.